Sammy Castleman. Oh, I'm going to restart now. I want to take the opportunity to introduce Sammy Castleman. She is a master's student in genetic counseling at UPenn here in the US. And this is one of her rotations. So last year we had Sam Miller from UPenn do a project for us. And this year, Sammy, we just happened to choose all genetic counselors that have the name Sam somewhere. So it just works out. And she's been lovely to have, and she's done a very thorough literature review of all the NARS1 cases that have been published. And so she's going to review that for us today. And then we're going to hopefully have an open discussion on how we can help better support you guys in the community. So, Sammy, please take it away.
Awesome. Well, thank you. I'm excited to be here, and it's been amazing getting to know Rachel and Kit throughout this whole process. So I just wanted to acknowledge that before I start and talk a little bit about, why I'm here, my love for science. I've always been interested since I was little, hence my picture in the lab here from undergrad. But the reason I love science is because I love working with families and communities in rare disease and elsewhere. And I'm really here because I'm inspired by all of you. And this is just a picture of my class on Rare Disease Day wearing our stripes. So, advocacy. And like Rachel mentioned, today I want to really review what progress has been made on NARS1 and research. And so I'm going to first talk about, from a broad level, what the timeline and path in terms of research really looks like to get to our ultimate goal of treatment or a cure for NARS1. And within that kind of section and overview, I'll go through the number of literature reviews I did, the number of cases I reviewed, and just kind of general demographic overviews. And then I'll deep dive into the actual literature, share some cases with you, review the symptoms that have been reported in the literature, and also symptoms that we don't see reported in the literature so much that we want to close that gap on and understand it better. And same with the cause of NARS1 as well. I will share what we've learned from the literature in terms of the cause of NARS1, and also share where we want to make progress moving forward. And just kind of off of that, my topic 3 will go into the Rare X updates. And then from there, we can go into that discussion of what you guys have run into, your experience with Rare X, and also just what's a priority to you moving forward in terms of research. So I'm excited. And to get started, we all share this same goal and mission of finding a treatment or a cure for NARS1. And when I set a big goal in my mind, I always think there's multiple steps to take to achieve that. So I've broken it down into multiple steps here. Step 1 being identifying patients with NARS1, which Rachel and this community has already done an incredible job with. And also, there's been incredible progress in the literature with that. And you can see that's kind of an ongoing progress, which is why the arrow goes across the screen. And then that leads into step 2 is once we identify an individual, we want to learn what symptoms they're experiencing, when they're experiencing those symptoms, what's causing those symptoms at a molecular or biology or genetic level, and how it might be similar to someone else with NARS1 diagnosis versus different from someone else with a NARS1 diagnosis. And that's really going to be the main part of my talk today, is going through what progress we've made in step 1 and step 2, and how that's going to impact step 3, which is figuring out what drugs and gene therapies will be critical to finding a cure or treatment for NARS1 going forward. And so to kind of start that deep dive, there has been so much progress made in terms of literature since NARS1 discovery in 2020. 12 publications is a lot. I've worked for a few different rare disease organizations and other organizations, and I've honestly not seen this many publications in this short of a time span. So I really want to acknowledge the progress that's made has been amazing. And from those 12 publications, there's been 56 NARS1 cases that I've reviewed as well. and 23 different genetic changes or variants at the gene or molecular level that have been identified. And this is a great step again to get to that final goal. And today I'm really going to be focusing on the literature that's on the childhood neurodevelopmental phenotype or symptoms. So that will be focusing on the right-hand side of this screen. You'll see there's 10 reports that really covered that childhood onset symptom NARS1 disorder and 45 cases. So again, I'll be focusing on those 45 cases. And just to kind of start with a broad overview of these cases that I've reviewed is they really cover a wide geographic diversity. North America, Europe, Asia, you see this broad coverage, which is really important and tells us NARS1 individuals are all over, and the more we can be inclusive, the more we can learn about the condition. And so I want to compare that real quickly into the patients in our community and all of you. And so there's more than just what the literature review says. And the purpose of this talk today is to say we've made so much progress. Let's continue. Let's get more people included and let's continue to learn. I think there is a jot form. Rachel, Kit, let me know if you want to pop that into the chat. And I think if there's any location that's not on here, you can share your contact information to be included.
I'll throw it in right now.
Awesome. And just continuing on this, demographic level, I wanted to quickly note that the cases I reviewed were split pretty equally between gender. So NARS1 can affect females just as often as it affects males. And you can see in terms of age range on the right as well. So this chart, each dot represents a case that I reviewed. The pink dots represent a case in which it was a female who was affected, and the blue dots represent cases in which there was a male that was affected. And then the y-axis is kind of that age range. And so you can see really from age 0 to 25, there's a variety of cases that were reported. And then there were fewer cases reported above age 25 on, what's the prognosis or what does it look like at an older age. And there needs to be more literature that's published at that stage is what this chart is telling us. So mostly what I'm reporting today covers childhood symptoms that are seen. Okay. And this next part is going to be that deep dive into the literature review. Again, this is like step 2 on that timeline of really understanding what symptoms make up NARS1. And so I'm going to start with 2 different types of cases that I reviewed and show you kind of what I've learned from those, and then all encompassing what I've learned about the symptoms of NARS1 from the literature, and also what's missing and where we need to make progress. So case 1, again, this is that age chart, and I'm zooming into one specific case report of a 5-year-old kid. And what I really want to emphasize with this case is that the symptoms that were reported were at one single moment in time. So the case report was actually written for this kid who was diagnosed with NARS1 after getting a genetic test complete at 5 years old. But the only symptomatic information this case provided was based on an evaluation done when this kid was 2.5 years old. The case reported symptoms like wide-based walking, spastic gait, which is kind of this jerky, uncoordinated movement, no expressive speech, and just challenges in terms of psychomotor development. Which it's true, but it's pretty general is the information, and it's still so important to our learning of NARS1, but it leaves us with a lot of questions and also doesn't cover the day-to-day experience that you all know from having and living and learning from people with the diagnosis. So there's a lot of questions here, and a lot more to learn. And so that's what kind of takes me into this case report number 2, which provides great longitudinal information and more of that diagnostic journey that's really helpful from a clinician standpoint to understand and to communicate to a family with the diagnosis what the prognosis is going to be moving forward, and where when you should be referred to certain doctors, or when you should step in and get treatment for certain things, or what treatment works and what treatment doesn't. These longitudinal studies give us more information as clinicians like that. And so this literature case number 2 is a male who was first reported in the literature at age 13, and it kind of goes back through his diagnostic journey of first having seizures at 4 months, sitting independently by 16 months, which is where they first started to notice this developmental delay of 16 months is usually later for sitting independently. And then it kind of skips like a bunch of years in this longitudinal review and goes to when he's age 13, and the kid at age 13 speaks 1 to 2 words, struggles with muscle challenges, and has microcephaly, which is the smaller head. And so, again, this information is great and makes so much progress in the literature, but there are a lot of questions that would only further help us communicate and counsel families with the NARS1 diagnosis. And so those questions are, what was the prenatal course like? Learning that information could potentially help us diagnose conditions earlier for families. What medication was he put on for seizures? Did that medication work? How, as a clinician, can I help get medication for seizures as fast as possible that work as fast as possible? And answering those types of questions are really important. Did physical therapy really help? At what age was physical therapy started? Is the seizure medication still working later on at age 13? Communication, was the AAC device really helpful? Having those things in the literature will only help make those types of interventions accessible to families and individuals with the diagnosis. And so we want to continue filling these gaps in the literature in. And so now those cases kind of show you, what types of cases are reported in the literature currently. And now we'll transition to like overview what symptoms have been reported in the literature. Again, I reviewed 45 cases. Here are the list of symptoms that came up in those cases. The first column here is representing any cases with the symptoms. The middle column explicitly means that the literature reported the cases did not have that symptom. And then the right column, which is really important to notify us where the gaps in literature is, shows that like the cases just didn't report on whether they had the symptom or not. So we're unsure about that case. And so I think developmental delay, which is at the top of this chart, which has been reported across all cases, is really well established as a symptom or something that's seen in individuals with NARS1. Same with microcephaly, which is that small head. Same with intellectual challenges. Same with speech delay. A lot of the kiddos, have difficulty speaking or speak 1 to 2 words or sentences or not at all. And so those are things that are well established in the literature. But then if you go down on this chart, you'll see other symptoms like challenges with eating, which have been reported in 2 individuals in the literature, but from my understanding is much more prevalent than that. And so there just isn't much data in the literature, and that's something where we as an organization can work to fill in and help understand, do we need to get individuals interventions for this? Are G tubes helpful? Things like that. Same with sleep apnea. Sleep is a common challenge for these individuals that's really underreported in the literature, and it'll be helpful to learn and understand what has helped these individuals with sleep, and what maybe hasn't helped, maybe something not to try. And so again, just kind of showing the differences between the top half of the chart, that there are some well-established symptoms, but there are also some less well-established symptoms that we're hoping to make progress on in research, and we're already making great progress with the Rare X data, which I'll go through at the end. And so this is just another kind of slide to highlight the same point of what's reported is more general right now and what we're hoping to do to enhance this step 2 of the research timeline and get into step 3 is just understanding in more detail the diagnostic journey of NARS1. And then I added a slide here as well, just on the management strategies, what gets at the point I was talking about with the second case, which is I'm curious, like what medications have worked for the individual for his seizures or what medications maybe haven't worked. And there's not too much data on the literature right now. So on the left-hand side of this slide, there were a few cases I reviewed that covered anesthesiology and undergoing procedures or BiPAP for sleep apnea. There's also a few reports of medications being used, but the report kind of stops after mentioning that it was used. It doesn't mention, did it work? Did it decrease the frequency of the seizures or did it get rid of the seizures completely? All of those questions are still out there for us to answer and for us to understand better moving forward. And so that takes me into this next step of the literature review, which is understanding the genetic cause for NARS1 disorder. And that's so important to understanding why the symptoms appear and why the symptoms might be variable from individual to individual. And so I want to start by just setting a base of why NARS1 is so essential and so important to us in our body for our bodies to grow and be healthy. And I like to introduce this topic by talking about an assembly line. And so here, this picture, we have an individual on the left-hand side, and the arrow points to an assembly line. And I like to think of this assembly line happening in each individual cell of our body. And so in each cell, there's an assembly line that is taking the NARS1 protein in our body and it's using it to create a tool, which is here in green, the tRNA. And that tool is really helpful and essential for building the proteins in our body that are used to help us grow and function. And so, for example, let's take a stomach cell. Let's say this assembly machine or assembly line is in that stomach cell. The NARS1 protein is building a tool that's helping our stomach cells digest the food we intake. And that's really essential to our body's functioning and digesting all the food we intake every day. Now, in individuals who have or are affected with NARS1, that protein or that assembly line can't function properly. So if we go back to this example of a stomach cell, the NARS1 protein in that stomach cell isn't functioning properly to create the tool here in green, the tRNA. And without that tool in that stomach cell, we can't build the proteins that digest our food. And that's why some individuals with NARS1 experience that indigestion or trouble in taking food and eating challenges. And that works similarly across all the cells in our body. So same with our brain cell. The assembly line in our brain cell, when individuals are affected with NARS1, can't do its assembly line job properly. And so we see symptoms like seizures, speaking difficulties, and smaller heads. And so that's happening from cell to cell in our body. And it's also important to recognize that there are different reasons that the assembly line in our cells isn't functioning properly. And those different reasons are because of different genetic changes in the NARS1 gene that are affecting the NARS1 protein in that assembly line differently. And so what I'm going to do next is I'm going to go through the variety of ways that assembly line can be affected. And why it's really important to understand that is because it will be helpful for this step 3 of understanding how do we repair that assembly line and what therapeutic options there are moving forward. What is the best one? And so I'm going to start by again, going through this assembly line in someone who doesn't have any genetic changes in their NARS1 gene. In these individuals, the NARS1 protein is fully functional and the amount of the NARS1 protein is at 100%. So it's at full capacity. And as a result, all of these tools are being made and being used for our body to grow. And so I want to kind of call out here that these barometers on the right side are both at 100%. The amount of protein is there, and the amount of function that that protein has is also at 100%. Now, when that assembly line has trouble or breaks down, there are a variety of ways, again, that assembly line can break down. And sometimes that line breaks down and it has a small impact. And other times that assembly breaks down and has a larger impact on how much of that tool we're making. And so this first example would be deep diving into what we've learned from the literature happens in individuals with one genetic change in the NARS1 gene. And for this example in particular, this individual has, again, one of the NARS1 proteins impacted, but it's not completely missing. So it's more like, I like to think of it like a sock. Like, let's say the sock is the NARS1 protein. Imagine there's a hole in the sock. Like the sock still exists, but there's a missing part of it or a different structure to it. And so for that reason, this barometer on the right side is at 100%. 100% of the protein is there, but only 50% or so of it is functional in making that final tool. And so when you have less of that final tool, there's less ability to make the proteins that you need for your body to function and do all the daily tasks that are really important. Now, another way this assembly line or machine could break down is one of the NARS1 proteins is completely missing or completely gone. In this case, the barometers on the right are showing that 50% of protein is there and that 50% of the protein is functional. And so similarly, you'll only have less of that final tool available to carry out your body's functions and to grow and to communicate. And then again, another way that this assembly machine or assembly line can break down or become unfunctional is to kind of have both of those situations combined, where you have one NARS1 protein that's missing or not there, and you have another NARS1 protein that's just missing a piece of it, that just has a hole in that sock. And so there's still some of that tool that's able to be produced, but it's not being produced at a high quantity to carry out these daily functional tasks of digesting food and communicating with your mind and your brain to speak and things like that. And so on the right here, I have, in this case, there's 50% of the protein, but even less than that is actually functional. And that's kind of important to note, these different variations or levels of functionality as I go into the next few slides, which will cover different types of gene therapies or treatment options based on knowing the different causes to the assembly line or different defects to the assembly line. And so the first is really going into the situation where an individual has one of those NARS1 proteins that are affected due to that missing piece or altered structure, so that sock with a hole in it. And in this case, the gene therapy would be actually correcting the non-functioning copy of the protein. So filling in that missing piece, sewing that hole in that sock together. And in this case, the gene therapy is really targeting like the function of the protein. So it's not affecting the amount of it there, but it's correcting the functioning. And as a result, the tools needed in the assembly line are made and the body can carry out the functions it needs for digestion and things like that. Now, in the next case, there's another strategy to gene therapy, which would be when one copy of the NARS1 protein is completely missing, but there is still one that remains, the gene therapy can actually boost production of the existing copy. And with boosting that function, again, more tools can be made and more protein can be made to carry out the body's essential tasks and essential functions like digestion and communication. And so it's affecting both barometers on the right here. And then lastly, in the last kind of slide I presented on how individuals with impacts to both NARS1 proteins have less amount of the protein and even less function. In that case, a gene therapy strategy could be to create synthetic NARS1 and add something to that assembly line that's not there yet. So adding that protein back in. And as a result, again, creating the tools as the final product and using that for communication, cell functions like communication, like digestion, and things like that. And then lastly, I just want to cover how gene therapy is really distinct. And in order to create a gene therapy, we need to really understand the cause of NARS1 at each cellular level. What's going on in this assembly machine versus drug repurposing, which is another thing that's really important that we're involved in, but it kind of targets the symptoms. So still, when we are using drug repurposing, at the cellular level, there are going to be those functional impacts to NARS1, but the drug will be treating symptoms like struggling to walk or pain or numbness, things like that. Those things that the drugs can potentially really help with in these kids. And so now that we've seen the assembly line, and the purpose of that is really to understand how critical it is for us as researchers to understand the cause of the defect or the lack of functionality in that assembly line. And that again goes back to what's going on at the gene level. What are the genetic variants? And so this slide is a picture of the NARS1 gene and all the literature I've reviewed showing which variants have been reported across the gene. And of course, like some variants are reported more than others, and some variants have more data than others. You can see here, I've kind of highlighted that there are good evidence for us to know what's going on in the assembly machine or assembly line for a good number of these variants, but there's also a good number of these variants where we don't quite know what's going on in the assembly line or at the variant level. And we want to learn more about what's going on so that we can figure out what treatment will be effective moving forward for many individuals. And so, now,
Sammy, can I pause you right there? And I'm
yeah,
just if anybody has questions, because that was a whole lot of
yeah,
information. Um, I'll pause
yeah.
just for a few seconds to see if anybody has any questions before we move into the rare X data.
When is the gene therapy should be going on?
Will you repeat the question?
The gene therapy, when are you guys gonna do gene therapy for the kids?
I mean, we're working on that. We're working on some strategies right now, but that takes years of development and investment for sure.
Would the insurance cover it or no?
That's a very complicated question that can definitely not be answered over Zoom.
Okay.
Yeah, and it's a great question as well. And I think it's really important to note that as we talk about these therapies, there are a lot of steps between step 2 and step 3 that need to occur. For example, like it's really important that we have samples donated to the biobank so that when we have an idea of drugs that can be repurposed or gene therapies, they can be tested in these samples first before going on to human clinical trials. So there are a bunch of smaller scientific steps that need to be complete as well. Any other questions? Or what other questions do you guys have? Yeah, Michelle.
Hi, sorry, I'm from the UK. So things may be a little bit different for myself. And just wondering with the actual these drugs that you're talking about, how many phases do you go through in regards to clinical trial? Do you go up to phase 4? Do you go from animal straight up to like controlled environment phases and then go into phase 4? Do you do all that the same as what we would here in the UK?
Yeah.
Uh, yeah, in the US, usually drugs are approved after phase 3, and phase 4 is considered post hoc or post approval. So we're
Right,
collecting data
okay.
right after the approval. So the EMA is very similar to FDA in terms of like, how safety and their regulatory processes work when it comes to new therapeutics. Um, it's also, again, we're a very, very, very small community. So again, I think how that is structured and how it will look is going to be different than a standard process because we're so small, right? So, right,
Yeah,
a lot of studies are based on, right, sample sizes and powers and, right, making sure you're getting adequate numbers, but
yeah,
we're so small, it's going to look different. So I think those are things we're also working on our side to really understand what that process looks like. There's a lot of work, or has been a lot of work in the US on that regard, and it's starting to also happen in the UK as well in partnership
right.
with the NHS. So I think there's some movement there in recognition that, the standard way of bringing therapeutics to market for ultra rare diseases cannot be applicable across all boards. But, how do we do that still safely and effectively? Um,
Yeah, exactly.
yeah. So, and then
Yeah.
for repurposing, obviously those drugs already exist, right? And so it's really,
Hmm.
so we're looking more at that symptom sort of management or quality of life improvement. And we
Okay.
have safety data, right? We have sort of toxicity data. We might even have some dosing information in children, and almost always in adults. So it's more of a, will that work for your child? What dose, do they tolerate it? That sort of thing. So it's a little bit of a different pathway that is, um, you know, less expensive and
Yeah.
simpler to do, right? It's for today. So I think
Yeah.
those are actually good questions and intro into Rare X, just because, right,
Okay.
because we are so small, and then
Yeah.
kind of how we can help, facilitate and change the conversation around, right, therapeutic development.
Of course. Thank you.
Yeah, thank you. Sorry, Sammy, I answered all those questions.
You're good.
You gotta cut me off sometimes.
You're good. Love the input. All right. And I can have time for questions at the end as well, or feel free to just interrupt me. There's only a few more slides before we get to the discussion section. But just a real quick, I know I covered a lot, but kind of like what Rachel was saying, we really want to start making progress and continuing to build our knowledge. And how we do that is through RareX. And so right now there have been 20 individuals who have enrolled, and they've enrolled from numerous different countries. You can see here, mostly the US and the UK, but we have numbers, across the globe. And what's really our goal is to get this number from 20 to 30. And there's steps to take between, 25 to 30 would be great, but we're really hoping to get to 30 individuals enrolled so that the more patients we know about, the more power, more understanding we have moving forward and can publish more data. And so here are just like some of the progress points so far. Here is a list of different symptoms that have been reported by individuals who have filled out the survey through RareX. And I think out of those 20 people who have enrolled, we've had 15 survey responses. So not only is it important for everyone to enroll in Rare X, but it's also important for surveys to be completed and to be completed often or regularly so we can have that symptom progression understanding as well. But here just highlights, 15 individuals who filled out the survey and 15 individuals that reported symptoms in the brain and nervous system. That's similar to the literature we saw where everyone reported developmental delay. But go down to digestive, for example, that was really underreported in the literature, but very clearly a prevalent symptomatic area for these families and individuals. And so that's a gap that Rare X is hopefully going to do great things and fill in, like does the G tube work? Does what diet has worked for individuals? There's certain things that I think could come out of that knowledge that could be really helpful moving forward for primary care doctors to understand, clinicians to understand, and also for diagnosis. I know for me as a genetic counselor, one of my jobs is I evaluate individuals who are coming into the clinic for maybe for developmental delay, and any additional symptoms we identify could help us get to the diagnosis faster. And so reporting these symptoms will help us get more individuals into the community as well, in addition to also helping their symptom management and prognosis moving forward. So in addition to digestive system, there's also pregnancy and delivery, which I think is an important area. Same with bones and connective tissue. There's not much on that in the literature. I think the literature reported 2 individuals with scoliosis, but not much on how severe that was. Do they wear braces? What has helped with that? Things like that. All questions we want to answer again. And so I know I talked about the digestive system quite a bit and our stomach cells. And so again, just showing how Rare X has already started to understand what the symptoms are like for these individuals in terms of the digestive system, feeding difficulties, and constipation being a really prevalent challenge that comes up. And so moving forward, we want to make progress in highlighting that area and helping physicians understand how to move forward or treat that area as well. And so I know, again, I've talked about a lot of different areas. I've talked about how in the literature we want to improve the geographic diversity that's represented, and we want to improve data on symptoms and management and therapeutic strategies, and also understand more genetic changes so that we can better understand the cause of NARS1. But what I want to discuss with you guys is, of course, any questions you have for me, but also what's a priority for you to have physicians know. I know, like, oftentimes we see families come in with binders of information, and I want to help educate the scientific community or the clinician community so that they're already educated by the time you come in. So, one, what's a priority to you? And also, what barriers do you face with Rare X, or concerns do you have with it? Addressing any of those so we can start to build up that population and fill in that gap. I guess also I'm curious, like, have any of you rolled in Rare X? Have you had a good experience with it so far?
I have.
Awesome.
I noticed my daughter, she has seizures, febrile seizures, and she has constipation. And
hmm
as the intellectual delay, which I checked her IQ is 57,
hmm
and the speech delay.
hmm
And I think the goal too here that we're hoping to accomplish is actually to publish our data on RareX so that we can fill in some of those gaps that Sammy alluded to in the literature. And I think even when I was just in London with, at Stephanie's lab there, you know, going around the room and kind of listening to everybody and having those conversations, like we recognize how much is missing. And so I think this is part of this conversation today is, you know, understanding what the priority is for you guys as caregivers and families on what you want, you know, again, the community to know outside of us, and then how do we, you know, get or uh, inspire others to enroll in Rare X so that we can publish it. So I think, how many did you have, Sammy? 45 individuals, right? So if
Yeah.
there's 45 individuals with NARS1, like we have right now 20 enrolled, we could easily, just as our little community, get to 45 or even more. We could have the largest publication of NARS1 impacted individuals, and that would be, you know, your and that's, you own that, right? That's all part of your contribution. So that's part of it. So interested in hearing some of your thoughts on all of those things and how we can do this better and what really matters.
And Rachel, maybe we can ask the question a slightly different way. Is there anybody who has not enrolled in RareX who's comfortable to kind of talk to maybe any concerns that they have or any difficulty that they've had? Because I guess we've communicated this a few times. We've also put a video now on our YouTube kind of showing the enrollment process. So if there is anybody who's kind of struggling, maybe we're missing something.
I think I've got your the little window open on my computer sort of all the time because I've always got lots of windows open, and I'm just knackered by the end of the day. So the prospect of filling in another form, I need to have a good day when the kids are left me alone and I don't have any other chores to do in the house. When I can sort of sit down in peace and quiet and fill in a questionnaire. So I think at the moment I'm just so tired.
No,
I
that's
think that's actually
fair.
really, really good feedback to get. And I get that it probably looks or feels a little daunting, especially when you see the data that comes out the other side. You kind of thinking, God, there must be a lot that goes in there. Just to bear in mind that the enrollment process itself is probably about 15 or 20 minutes, and there's no requirement to kind of suddenly do all of the surveys waiting for you.
Yeah.
You can just enroll and do nothing else, and then, you know, come back in a couple of weeks and just do one survey or even a half of a survey. So certainly don't feel under pressure to, you know, that you know, you have to do every survey that's in there within, you know, a week of enrolling or anything like that.
And I think that's the other sort of point too, is, you know, what is really a priority for our community, because there's so many surveys in there. And I have been speaking with RareX too, and they're trying to streamline that and make it more disorder specific, so it's not so overwhelming. But I think that's also part of this conversation, is if there's, you know, certain surveys like sleep surveys, right, that, right, it's like, you know, all of our kids struggle with, or a lot of the kids struggle with, right, or some of the GI stuff, you know, that sort of stuff. Those are the things that we want to make sure that we're, you know, communicating to you all, like, hey, don't worry about the rest of these surveys. Here's the ones we really want to understand. And so, Sammy, if you go back, can you go back to your slide where you've separated that out in the literature, some of the,
Mm-hmm.
yeah. So like even here, like if there's some, or go all the way back down, go back up. Yeah, there we go. Like this one, like if there's ones here that like resonate with you, like symptoms that resonate with you that are just missing or sparsely reported, those are the things that we want to make sure that we're, you know, helping to support you. Because yeah, I mean, I've enrolled, I've filled it out, I look at it, and it's a lot when you go into the dashboards, when you're working and you're managing kids and all of the other stuff. So that's what part of this conversation is for too.
Can I add something?
Yes, please do, Nancy. Having a lifetime of.
I mean, I had help with it. as you
Yeah.
know, probably Lauren, but it was hard. I'm one of the ones that has a 33-year-old, so it was hard to look back. I think I answered them as well as I could. He had a lot of these symptoms other than he does not have seizures. So it is tough to remember when you're not in it like the most of you are. But I'm probably speaking just for myself. A priority to me as he gets older is, what does this disease look like for James? His life expectancy, his motor ability. I could say 2 months ago he could barely walk. It's up and down. What's the progression? But I don't want to make it all about me, but that's where I'm at, age-wise. What can I expect for him? What kind of life does he have the next 33 years?
I think everybody wants to know that answer too.
Yeah, I know. We want that crystal ball, Rachel.
I know. I know.
No, but I mean, definitely his gross motor has dramatically changed. If I, go back, and it took him so long to walk, and, he was actually running as the adolescent, and to think that he has to hold on to things as he walks, and can't walk without AFOs. It's just one of our concerns, but I know you're not going to have an answer for me.
No, and I think what you're speaking to, Nancy, speaks to some of the stuff on Sammy's slide related to the hyperreflexia and the peripheral neuropathy, right? Because, it's not like, especially the neuropathy issue. I mean, there's twice as many that it's not even reported in. And so I think that's something that, all of us, we talked about this in London too, is we need to understand this aspect of it, and how is it contributing to their walking and their independence. And so, we have a few older young adults in our community, and so your contribution is huge, and we want to encourage as many as we can get, right, as they get diagnosed later in age, to continue to do that. But this neuropathy issue is something, again, can we start closing the gap so that we can understand that as early as possible? Same way with the hyperreflexia, which is some of that spasticity and extra movements, or things that really are affecting the coordination of the kids too.
What's your gut feeling that I have to think there's a lot of older people with this.
Yep.
You know what I mean? And I pursued this because of my daughters, and was there and being pushed, living in Boston and having them push for a diagnosis. But I have to think there's probably a significant population that has not been diagnosed.
Yep. I suspect, this is true. Sammy, you can speak, you could probably know the stats. Do you know the stats around the undiagnosed populations of various disorders?
I don't know the stats, but like this first case here, I think is a great example of this 5-year-old kid was diagnosed with cerebral palsy, and it diagnosed at age like 14 months, I believe. And then it wasn't until they were 5 years old where the parents were interested in exploring genetic testing, and they got diagnosed with this NARS1 disorder from that genetic test. But the child could have gone their whole life and with the diagnosis of cerebral palsy. And so if you think back to how many individuals have cerebral palsy, which is, a more prevalent disorder and more common, how many of those individuals haven't gotten genetic testing? It's probably numerous. And so what are we missing there in that population is a question that comes to my mind.
James was also put under that umbrella. So I see that. And it wasn't for the neurologist when he was a teenager saying, let's do this nerve study, and realizing he had severe nerve damage. The gene geneticists were saying you're going to get a diagnosis based on that more than his, developmental delay. And they were absolutely right. So thank you.
My son's been diagnosed with severe autism That was what he was first diagnosed with when he was 3. Um, Obviously, I didn't feel like that was all it was. So the further along we got with his age, he's just about turning 5, but in the past 18 months, there's been a massive change in how much he can do mobility wise, especially as he's getting a little bit older. This became a real concern from his pediatric, and one of my questions when I seen that the NARS gene there was a variant in his NARS1 gene was all the behavioral and emotional aspects, and I was a huge part of me was thinking how many children have been maybe misdiagnosed with autism as well, because they put global developmental delay and mobility issues or mobility impairment, for instance, with autism, especially in severe autism, that that could be another huge misdiagnosis, I feel. And obviously I take from that is that there is many out there that have been diagnosed with severe, do have these impairments, who could have NARS1, that have grown to live an independent, happy life. And that's, as we haven't got any idea of a life expectancy. I have no idea. I've only just been informed of this 4 days ago. So I'm very, very, very new to it. But all these thoughts have come to my head in those 4 days. It's a little bit of it's a bit of breathing space, should I say? Knowing that it could just carry on. With just the correct support in the right areas. And I did want to ask a question. I missed the beginning of this, so I do apologise. But can I get more information on the enrolment of RareX? Is it just trying to pick up the literature of the symptoms and the daily life of the children that potentially have it? Is that all it is, or is it?
It's basically a parent entered survey system. And so, yeah, it's essentially, it walks through, it'll walk you through, your child's life, right? And actually including your pregnancy. And so even when we see from our data now that there were some maybe signals somewhere in pregnancy that there could have been something, right, that was happening even in development. And so those again are all things, because that will improve sort of diagnosis, earlier on. And so it walks through the journey of your life, your child's life. There's also, right, medication histories like supplements, diets, right, that might be helpful or useful, because that can also help inform us as we start thinking about not just publishing in the literature, but once you can publish in the literature, this also helps us form standard of care, right? And so, and then it is right. Are these kids getting EMGs? Are they getting standard MRIs? When you get MRIs, it can kind of tell. It might be able to provide some insight to the brain and what other associations can be happening because of abnormalities in the brain from the beginning. So there's lots of things that it can do. Right. If everybody's struggling with feeding, right, G tubes earlier, or a certain diet perhaps, right, that sort of thing, to help with weight gain and maybe even seizure management for some of us, depending on the child. So that's really the intent of it. Ross, did I miss anything on that you want to say?
No, I was just going to suggest that we can just set up a kind of a one-to-one call as well. I'm conscious. Did you say you got the diagnosis 4 days ago?
Yeah, 4 days ago. Yeah. She's
so
Yep. She
Yeah,
just came through our system, through our email, a
yeah.
couple of days ago. Yeah.
And look, what I will say is, for us with Keane, I think we're about 3 years. That first period after you get the diagnosis is obviously, that's a tough time, right? And I kind of feel for you that you've been dumped a lot of this information here this evening. So I'd suggest maybe we kind of take it to the side and we can kind of set up a smaller call with myself, Terry, and Rachel, and we can probably take you through this a little bit more from the beginning.
Yeah, that would be great. I just kind of Rachel give me the option
Yeah,
to be able to join this, so just so I've got a bit more of just some form of clarity, really, because there's nothing here at all, is there really, to help with our
yeah,
information. Um,
yeah,
and like you said, there's so many questions, so it's just nice to have a bit of an
yeah,
understanding of this and other people's perspectives and what they're going through.
Yeah.
yeah,
And we're hoping that, again, this provides a resource, not just for you guys as parents, right? But also, right, this is something you can share with clinicians and, other
Yeah.
people, right? As you continue to see them, to say like, no, this is like a real thing, right? Like,
Yeah.
so I think all of that is the reason why we wanted Sammy to do this, and to also make sure that we can collect and collate the information, from you guys as families, and to, supplement the knowledge, but really to also to really fill in the gaps and get the care that, all these kids need, and to understand, Nancy, the progression, to understand what James's life looks like, right? And all these other kids, and because we really don't know the answer to that. So I do get very excited when we get 30-year-olds. I'm not gonna lie. And
So
it's
look,
been,
I think if anybody on the RareX data, again, if anybody wants to spend a little bit of time and go through this on a kind of a one-to-one Zoom session, just reach out and drop me an email, Ross at the Rory Bell Foundation.org, and I'll set up some time that's convenient to you to go through and get some of this information in. And Michelle, I guess just to you, one other point is there's lots of terminology in here which I didn't understand for the first 18 months. So there's lots of science in here, but we will help you to kind of break it down and make sense of it. And there are things that affect the different kids and different adults differently, I guess, just to take that away.
Yeah.
And because I know, and I just say that from our own experience, we sort of almost took the worst of everything, put it together, and said that's what we're getting, and that's kind of not how this works. So take a breath, and we will catch up with you separately.
Thank you.
Ross, she
Thank you.
already knows what de novo means, so she's like,
I did used to work in clinical research, so I have a tiny little bit of understanding of
perfect.
certain things. But obviously, this is still super new, super fresh, super scary, terrifying. I don't want to close my eyes and see him, like waste any time without him. So, I'm very appreciative of you offering the one-on-one so I can get more understanding for my son.
Sure, no problem. Sorry, Sammy, back to you or to Rachel.
We're good. Thank you for filling in. Super helpful.
Are there any, we're right at time. Are there any other questions or comments, thoughts, before we close out today?
Hi, everybody. What what about RareX in other countries outside the US? How can, for example, for French or other European or any other country feel RareX as it's exclusively in English?
Yeah,
How
we're
we
still,
can,
yeah, we're
how we
still,
can translate and explain to other people how they
yeah,
can.
yeah, I would love any help on that. We are still working with them and trying to get these translation services up, with RareX. So, where the surveys will be translated. I think some of it has been a lot of it has been licensing issues, unfortunately, with the specific surveys.
Because there's two problem, language problem in addition to the scientist one. So
Yes, yeah. But I also like Ingrid, I know, one of our researchers at Scripps is happy to like hold an all French section if we need to, because I know we do have probably 5 or 6 families in France that have cited the language barrier and being able to do this and complete it. And so
Hey,
that's
Rachel, can I
one
interrupt
way.
you?
Yes.
When we first rolled out Rare X, didn't we have someone create a French language introductory video?
I don't think we did. I don't think we did.
Okay.
But I think that was something we had talked about, and Ingrid would can do that and will do that.
Okay.
It's more of though, once you get into the surveys though, is reading it, right?
Yeah. And are they have any languages other than English currently? I thought there were a couple, but no.
No, the only one that will go to Spanish is the Vineland, because Pearson, who owns the Vineland, has done that. The rest are in English only, and it continues to be a pain point for many of us.
Gotcha.
So.
And Chris thinks that Terry's cousin may have done something in French when we originally went live. So maybe more to come on that.
Right.
If we have
Yeah.
something, we can send it out for sure.
Yeah. And I will keep on harassing Rare X too about where's the language translation thing. We've been talking to them for quite some time on that issue.
Fauzi, when you use Google Translate, obviously doesn't assist you there, does it? If you do the full website translation.
Yeah, but it's not too efficient because many scientists' words are not translated very well in Google Translate. So you can use ChatGPT, for example, but yeah, even AI, it's not enough performance. So as a physician, I find it sometimes difficult. So I guess that someone else, can really find it hard. So I think
And again, I think if you do come across any tooling that you think we can invest in as an organization, bring that to us, because we'll go out there and ask people. But I guess we don't use these types of solutions ourselves. So if you have something to bring to us, by all means, flag any service or that you think we can invest in.
Yeah, why not?
And that's something I can also check in with any of my colleagues on and see if they, in their experience with Rare Acts at other organizations, if they've come across anything that's helped there.
And I think it's just even any tools, because I've tried Google Translate before, and you're right, Fauzi, it doesn't.
Yeah.
It doesn't do a great job, and I have found that even with communicating with our families who speak different languages, I often have to put it through Translate like a couple of times and rework the language just so that it fits right or sounds right. Yeah.
If there is, one of the things that we can always do is go back to the old analog model. And if there are people who really are invested in doing this and do not, the translation services aren't working, we as an organization can look into investing in interpretation services to actually be on the call and fill out the forms with them. Potentially, yeah.
At least the most important ones, especially like that initial head to toe, which really starts to like get at not only the symptoms that are appearing across the body systems, but also when they start appearing. which again is another really important piece of information that Sammy highlighted, right? It's that point of time versus like what happens through the trajectory. And so it's really important for us to understand in terms of like, right, how all of the children age, right? How that's impacting them. So that's really one of the biggest ones that we can do. All right, I know we're at time here. Any other questions before we hop off? I want to thank Sammy for her efforts. This will be posted on our YouTube. We'll have our slide decks available. Sammy also is going to try to help us hopefully this summer, if we can get some more folks in RareX at like mining that data and helping to get a publication out. So that will also be super helpful for us. So we'll try to work and solve some of these problems that we heard today and try to figure out how to streamline the survey number, find some translation, either services or tools that might help, and then we can go from there. Does that sound reasonable? All right. Any other questions? You guys know how to get a hold of us or join our WhatsApp or whatever, and we'll happy to continue this conversation.
Thank you, Sammy, for your work on this.
Yeah. Thank you, everyone. Nice
Thank you
to
so
meet all
much.
of you.
Thank you, Sammy.
Thank you.
Thanks very much.
Thank you.